good evening around the world. All the subscribers joining today's webinar. I first wish you all very happy new year. This is our first webinar for the season of this year 2023 spring semester. I am Dil Thavaraja, professor at the Clemson University. I oversee Pulse Quality and Nutritional Breeding Program at the Clemson University. We develop Pulse cultivars, peas, lentils, chickpeas with high nutrition quality for organic cropping system. First, I would like to thank our funders, USDA, NIFA, National Institute of Food and Agriculture, USAID, AID, Feed the Future Innovation Lab, South Carolina Department of Agriculture, the Good Food Institute, USDA Agriculture Service, ARS, and the Global Food Shop. Today we have a very special speaker. He is actually my master's supervisor, a professor who taught me soil science and how it impacts the food system, and which makes my program shape up with his wisdom that he taught me. Dr. Jeff Schoenau. Dr. Jeff Schoenau is a professor of soil fertility and professional agrologist, University of Saskatchewan, Saskatoon, Saskatchewan, Canada. He is also the Ministry of Agriculture Strategic Research Program Chair in Soil Nutrient Management. Dr. Schoenau is a professor of soil fertility, and he has been teaching growers, students, researchers like me around more than 2-3 decades now. He holds the Ministry of Agriculture Soil Nutrient Management position, and also his research is focused on practices that provide better plant nutrition and improve soil quality prairie cropping system in Saskatchewan, Canada. One of the key things, and one of the things that I love about Dr. Schoenau, he is the farmer as well as a full-time professor. It was eye-opening how you can actually connect your researchers into your own family farm. His spouse, Miss Leanne, near Central Butte. I also visited his farm in 1999, long time ago, and he grows peas, lentils, wheat, and canola. Thank you, Dr. Schoenau, joining with us today, and it's my pleasure to have you in our webinar series.
Great. Thank you very much, Dil, for that wonderful introduction, and I've really enjoyed when you were a student and interacting with you over all these many years. So thank you very much, and thank you for the opportunity to come out and share a few thoughts from the Northern Great Plains about pulse production and soils and fertility. So I'm just going to share my screen here and grab my PowerPoint. Bring that up, and everybody can see that, I think. Great.
Yes.
Okay.
Yes, we can see. Thank you, Dr. Schoenau.
Excellent. So yes, I really appreciate this opportunity to share some thoughts with you folks about soil fertility management and soil health as it relates to organic pulse crop production. And over the years, I've been fortunate to work with a number of different crops in my soil fertility research, and I've always really enjoyed working with the pulse crops from a research standpoint. Very, very important. Do good things for the soil, as I'll talk a little bit about, and also I grow pulse crops on my farm, as Dil said, peas, as well as I've grown chickpeas and lentils and faba beans and a host of different pulse crops. And I think that's great that in South Carolina, looking very with a lot of interest in developing pulse crops in organic production systems, as Dil tells me, primarily as a winter crop. A little bit about where I'm coming from in the province of Saskatchewan in Canada. We're a little bit more limited in our environment than you folks are in South Carolina. We grow one crop per year, spring seeded, and we only have a 4-month growing season that runs from about May until September. And we're pretty dry as well. We're a semi-arid environment with only about 400 millimeters of annual precipitation. Our soils, as shown there, were developed under under grassland. So they're what we call Chernozems. In the southwestern portion of our province, we have dry brown Chernozems, quite similar to the soils you'd find in Montana and Wyoming. As we move northeast across our province, gets a little bit wetter. We get into some thicker black Chernozems or mollisols. And then finally up the top there, we have our gray soils. And those are soils that were formed under boreal forest. And actually, we're fortunate we're able to grow pulse crops in all the different soil climatic regions of Saskatchewan. And they are an important part of our crop rotation. Just to put things in perspective, in Saskatchewan, the University of Saskatchewan, where I work at, is located in Saskatoon. And then about 100 miles south of there, where the blue dot is, that's where my farm is at Central Butte. So it's on the boundary between what we call the dark brown and the black soil zone. And in that area, we grow a wide variety of different pulse crops, including peas, lentils, chickpeas. We're a little bit on the dry side for the faba beans, which tend to be grown in the more moist areas like Saskatoon and further north. Over there on the left-hand side, that's my farmhouse on my farm. And on my farm, which is about 1,600 acres, I use a lot of my land area for research projects that are conducted by graduate students and research associates. It's a farm that has been in my family for over 100 years. It was brought under cultivation, native prairie, back at in the early 1900s. And in terms of legumes, I guess one of the crops that's actually continuing to expand and moving into our prairie area is actually soybeans, as a result of the advent of some short season varieties that are seeming to be relatively well adapted to our very short growing season. But dominantly there, is shown on the bottom, a rotation I have on my farm that's commonly used is canola, and then a pulse crop in the middle is shown there, like peas or lentil, and then a cereal crop like wheat. And we have to take some pretty important measures out there to conserve moisture. And so actually there on the right shows one of my field experiments where we have treatments of different stubble height in order to trap snow to get some additional moisture stored in our soil profile, because we rely on stored soil moisture in our soil profiles along with growing season precipitation to get us enough moisture to get through the season, folks. So that's the kind of cropping system that we have, and that's the environment that we do our research work in. So to talk a little bit about pulse crops and fertility and some of the things that we've done over the years and learned, I guess the first one, of course, very important is nitrogen, and really it all starts there, is shown there in the nodules on the roots of that chickpea plant there. And of course, those nodules are very important as the home for the rhizobium bacteria that carries out nitrogen fixation, fixing nitrogen for that legume crop, and also contributing to the nitrogen nutrition of the following crop as well. So that process that we call biological fixation of atmospheric nitrogen, key component of a pulse crop, and particularly when it comes to the influence of pulse crops on soil fertility in an organic cropping system, as we'll see. So the nitrogen fixation process by legumes, just to give a quick overview of that, really is what we call a symbiosis or a two-way street. Both partners benefit. That plant sends energy in the form of photosynthate to the nodules, and within that nodule, we've got that rhizobium bacteria. And if we've inoculated that seed or soil with a superior commercial nitrogen-fixing strain of rhizobia, we've got the ideal conditions then set up for that rhizobia to use that energy from the plant and convert atmospheric N2 to plant usable nitrogen using the nitrogenase enzyme in that nodule. And so the rhizobium gets energy, and in turn what it does is it sends that fixed nitrogen to the plant. So it's a symbiosis, and It is an energy expensive process, folks, and there is a cost to the plant. The plant has to give up some energy in order to get that fixation and get that nitrogen from the rhizobia in that nodule. So I think always a good rule of thumb I've kept in mind is if something's going to affect the plant growth, it's probably going to affect the fixation contribution. So I guess a question folks often ask, how much nitrogen is derived from the air and converted into plant nitrogen by a legume crop? And it is variables. There's different ways to measure it that give different numbers. We use a N15 isotope dilution technique. It's one approach. But any way you slice it, it really ends up being a pretty significant, economically important contribution. And as a good rule of thumb, I always remember about 3/4 of the nitrogen in a pulse crop comes from the atmosphere via biological fixation under good conditions in that nodule. And we'll talk a little bit about those conditions in just a moment. But yeah, we can get 3/4, even more of that nitrogen in that plant coming from the air through that fixation process. And when we look at the roots of those pulse crops and we look at those nodules, and as shown there, if we cut them open, if we see a really bright red color, that red color indicates the presence of leg hemoglobin. Leg hemoglobin, red color, kind of like the hemoglobin in your blood, and that red color tells you that that nodule is actively fixing nitrogen. The leg hemoglobin shields that nitrogenase enzyme from oxygen and allows it to work to fix nitrogen in those nodules and then make it available for the plant. In Western Canada, these are the typical amounts of nitrogen we get from fixation. And I think these are going to be less than what you folks in the southeastern US would typically achieve, because our environment, again, is very much limited by a very short season and dry conditions. But I think the relative amounts here would probably still be applicable in South Carolina. The forage legumes like alfalfa tend to be on the top of the heap when it comes to nitrogen fixers, but not far below that is faba bean. And actually, faba bean is an annual legume that can really fix a lot of nitrogen. Typically, it's the highest annual legume or pulse crop in the fixation scheme. Below that, peas are good nitrogen fixers. Soybeans, we've found actually when we inoculate them with a superior commercial inoculant, can actually get really quite good fixation out of soybean. Lentils and chickpeas, also good fixers. They're a little bit lower there because they tend to be growing in the drier regions of our province. And finally, dry bean, it's the one that maybe isn't as good a nitrogen fixer as the rest. And part of the reason is we really don't have a good inoculant for that dry bean that can really boop that nitrogen fixation process up. How much you get, of course, is going to depend on a lot of factors. Inoculation, nodulation, environmental conditions are important. Anything that affects growth is going to be affecting that nitrogen fixation. Soil available nitrogen, as we'll talk about in just a moment. If you've got lots of available nitrogen in the soil, that will hinder the nitrogen fixation process because the plant will just use the available nitrogen in the soil instead of fixing it because it costs it less in energy to just use the available N in the soil. And of course, other nutrients like phosphorus are also important. Again, a well-nourished plant in terms of a pulse crop is one that will also have optimal fixation. And what we find is that when we look at nitrogen balances in this pulse system, the amount of nitrogen that's fixed by that pulse crop can often approach or even exceed that which is removed in harvest. And that's a really good thing in terms of maintaining fertility, because the nitrogen that's removed in that crop harvest is coming from the air through biological nitrogen fixation in that pulse crop, instead of coming from the soil or having to come from fertilizer. As I said, growing pulses on soils where you've got high levels of available N, like a lot of nitrate nitrogen that may be present there, that's probably not a typical situation in South Carolina because of your environment. But in the semi-arid environment of Saskatchewan, we can end up with a lot of residual nitrate in the soil if the crop happened to not use the fertilizer that year. And if we grow a pulse on that, that will reduce the contribution from atmospheric fixation. And a couple of pictures that I got there that I took from a demonstration I do for my class show that quite nicely. With yellow peas there, they were inoculated, no nitrogen added on a nitrogen deficient soil. You can see there on the roots there, the little nodules that are present there, and they have a reddish kind of color, actively fixing nitrogen. Whereas over there on the right-hand side, these are the same peas, inoculated, but lots of nitrogen fertilizer added there, 200 kilograms of N per hectare, and there are no nodules on the roots there. Very little or no nitrogen fixation taking place because the peas are simply using the available nitrogen in the soil. They're not sending energy to nodules. They're not allowing those nodules to become established because it's energetically more efficient for them to simply use that available N in the soil. So if you want the maximum contribution for biological nitrogen fixation, folks, grow those legumes on soils that are low in available N. So we talked a lot about nitrogen fixation. I guess another question that comes up, very important when you have those pulse crops in rotation, what's the nitrogen contribution of the pulse to a following crop? And I say there, it's significant, but it's not typically huge if the seed is harvested and removed. Now, if you're taking that pulse crop, that legume, and plowing it all under the entire plant as a green manure, then your contribution of N is going to be a lot higher. But remember, when you take that seed off in harvest, it contains about 70% of the nitrogen in that mature crop. So when it's harvested, there's not a huge amount left behind. And just to kind of put some numbers to this, in a simple example, a pea crop there, 35 bushels per acre, 140 pounds of N per acre in that crop, 70% derived from fixation. Just to make the numbers round, it works out to about 100 pounds of N per acre from fixation. Now, we know that harvest also removes about 70% of nitrogen in that plant. So you got 100 pounds of nitrogen per acre leaving the system in the harvested grain. So where you see the benefit in this system is that the nitrogen that's leaving that soil, that system in the harvested grain, is just about equal to the amount of nitrogen that's fixed from the atmosphere. So essentially what you're getting is nitrogen in that harvested pulse seed obtained for free from that biological fixation of atmospheric nitrogen. And on the other hand, if it's a non-legume, that nitrogen has to come from the soil and fertilizer. So that biological nitrogen fixation works very effectively in an organic system to help maintain that nitrogen fertility in that soil over the long term without reliance on external fertilizer nitrogen inputs. So we talked about this, but again, what about next year's crop? Where do the benefits come from? And we think about benefits from pulses in rotation, it's probably worthwhile to start out with a non-nitrogen benefit, because often that non-nitrogen benefit is as big as the nitrogen benefit itself, particularly when the grain is harvested off the system. So that non-nitrogen benefit can include things like legume pulse in rotation, breaking pest cycles, certainly improving the soil tilth or structure. Also, soil moisture utilization. We find here in Western Canada, pulse crops like peas are shallow rooted, so they leave behind some deep moisture for the following cereal crop that we typically grow in rotation. Also, they promote beneficial soil biological activity. Organisms like arbuscular mycorrhizal fungi, PGPR, are promoted in rotations that contain that pulse crop, and also promote the uptake of other nutrients as well. And this non-nitrogen benefit shows, I think, very clearly in this figure here from some work that was done in Saskatchewan, where barley was growing on barley stubble versus barley growing on a pulse stubble like faba bean, field pea, and lentil. And what you can see there is that in this experiment here, adding nitrogen fertilizer to barley growing on barley stubble could never, ever allow that barley yield of the barley growing on barley stubble to achieve the yield of the barley growing on the pulse stubble. So what you see there is even adding nitrogen fertilizer to overcome some of the direct nitrogen benefit from the pulse crop, you're still never achieving the same yield from the barley on its own stubble compared to growing on a pulse stubble. And so that difference there is what we can see there and refer to as the non-nitrogen benefit. And sulfur pulses, we typically observe in our environment about 4 to 5 pounds of plant nitrogen there in that pulse crop per bushel of grain yield that we get. And again, remembering that about 75% of that N comes from fixation. And again, when we harvest that plant, about 75% of that above ground plant nitrogen is removed in the grain. And actually, we find that if we think about where that nitrogen is distributed in that pulse crop and ultimately becoming available for a following crop, we find that the nitrogen that's contained in the above ground straw actually makes relatively little contribution to nitrogen supply to the following crop. And generally, less than 10% of the nitrogen in that straw, that above ground pulse residue, is made available to the crop in the following year, folks. In fact, some work that one of my students did in controlled environments showed only about 4% of the above ground P residue N, only 3% of the above ground soybean residue N was actually recovered by the wheat. So the direct nitrogen contribution to a following crop from that pulse crop really doesn't come from the above ground material. Instead, folks, we find that the majority of that contribution comes below ground through decomposition of the old legume roots and nodules that starts to take place in the fall and proceeds into the following spring and over the following growing season. We were also interested in some of the research work that we've recently done in the ability of different pulse crops to contribute different amounts of available nitrogen as those residues decomposed in the following season if you were growing a wheat crop. And so how we did this is we grew wheat crop on different pulse stubbles, and we also used probes we call PRS, plant root simulator probes, which absorb the nitrate and ammonium, the available nitrogen that's released over the season as those residues decompose. And what we found in this work, and this was at a couple of sites around Saskatoon, Saskatchewan, around the University of Saskatchewan, is that actually, folks, there wasn't a huge difference in the release of available nitrogen from the different grain legumes, soy, pea, and lentil, but generally is shown there, pea and lentil, especially at the beginning of the season, did tend to be a little bit better in the amount of available nitrogen that was released in the soil from their residues compared to the soybean. And compared to wheat, the release of nitrogen from all of those residues of the pulse crops and the soybean over the season were much greater release from the legumes compared to the wheat, as we would expect. And I think an interesting thing, and we've observed this in our studies as well as others have, is that crop nitrogen uptake by a crop grown on pulse stubble is often higher than what is explained by the direct nitrogen contribution from the above and below ground residue itself. And I think this can be attributed to overall just some other things going on, like better conditions for root growth, stimulated biological activity, and I guess for lack of a better word, it's what I often refer to as the magic of legumes. So that's nitrogen. What about phosphorus? Well, legumes do have high phosphorus requirements because that biological nitrogen fixation process requires lots of energy. And we know that phosphorus is the energy currency of life. Now, unfortunately, we don't have phosphorus in the air that plants can use. For phosphorus, all of that phosphorus has to come from soil and fertilizer, or some other type of amendment that adds phosphorus to the soil. And lentil, pea, chickpea, faba bean, as a general rule of thumb, require around a pound of P2O5 per bushel of grain that's grown. And like nitrogen, about 3/4 of the phosphorus in that mature pulse is in the seed, and that's removed in harvest. Now, the thing about legumes when it comes to phosphorus in the soil is legumes, pulse crops, are very good scavengers of phosphorus as well as other nutrients like potassium and sulfur because they develop this strong mycorrhizal relationship. They have a high infection of this beneficial AM fungi on their roots, as shown there in the picture in that photo that shows that AM fungal hyphae extending away from the root there, and that AM fungal hyphae does a great job of increasing the surface area of the roots, just extending that rooting system and allowing it to do a better job of getting especially immobile nutrients like phosphorus and potassium out of the soil, but also other nutrients as well. So legumes are real good scavengers. They can often mobilize and access phosphorus, we find, better than a lot of other crops can. And that, I think, explains why I've done a lot of phosphorus fertility trials, response trials with pulses over the years, and I've typically found them to be the least responsive to pea fertilization compared to oil seeds and cereals. And that relates back again to them being good scavengers and that mycorrhizal association, that strong mycorrhizal association that develops with the roots of those pulse crops. Thing about the pulse crops is in terms of needs for phosphorus and when they need to have it available to them, they need it and take it up early on in their growth. Some work that we did a few years ago with pea and lentils showed the maximum accumulation rate of phosphorus occurred quite early on in the growth cycle, about the branching period, and that would be the place where the slope of that curve is the steepest there. You can see for pea and lentil around about 1 month or so after emergence. That's our time when we have the most rapid or highest rates of phosphorus uptake. And so you want to make sure that the crops are going to be able to access that phosphorus early on. And that's why as well that we want to have if we're putting fertilizer or some type of other amendment onto the soil as a phosphorus source, we want to make sure that the roots of that germinating seed and seedling can access it early on in the growth cycle. And that in turn promotes good growth and what we call the pop-up effect that we see from a well-nourished pulse crop that has ample supplies of available phosphorus. So that's the pulse crop itself. What are the effects of pulse crops on the phosphorus nutrition of a following crop? And here I go back to some research work that was done by another graduate student that worked with me, Catherine Saluski. And what she found is that pea uptake by a non-legume like a cereal or an oil seed was generally increased when it followed a legume versus a non-legume. And some possible reasons for this include enhanced recycling of phosphorus from the residue itself. I think a very important one is the increased colonization of roots by this beneficial AM fungi that we talked a little bit about earlier, and also just overall improved root growth, health, and nutrient demand by the following crop when it's growing on a pulse stubble compared to, say, its own stubble. So those are some factors that we identified. That increase in uptake we found really didn't relate back to any kind of an increase in the content of available phosphorus in the soil, but instead it comes back just to the ability of that legume growing the year before to make that soil better. I would say healthier, for lack of a better term, to access the nutrients that are in that soil, and that really involves modifying that rhizosphere, that root zone, a better rooting environment, improved root and soil health, and again, coming back as shown there, that AM fungal relationship, very, very important in improving the ability of those roots to get those immobile nutrients, insoluble nutrients like phosphorus out of that soil. Folks, that's a benefit that's kind of difficult to directly measure. But I think some of the new soil health tests that we see out there, I think can provide insight into this, particularly the ones that are looking at microbial activity, enzyme activity like phosphatase in the soil. I think are some of the measurements we can make that can provide some further insight into what those legumes, those pulse crops are doing as far as soil improvement is concerned. The last thing that I'd like to talk a little bit about in the few minutes that I have remaining is some work that we have going on right now in Western Canada, and it's going on in other regions of the Northern Plains, and certainly I think in North America throughout the world, and that's intercropping. And I want to emphasize here particularly pulse oilseed intercropping. And what we're talking about here is growing pulse crops together with oil seeds. As shown there in the photo, one intercrop system that we've been working with is a chickpea flax intercrop. And in this case, shown in the picture is a chickpea flax alternate row intercrop, where we're growing flax in one row, and then right beside it, we've got the chickpea, and alternating rows across the land area. So we've got those two crops then growing at the same time as in alternate rows. And the other system that we look at and have been looking at in our research work, the other configuration, as shown here with a pea mustard intercrop as a pulse oilseed intercrop, is a mixed row system where we have both the pea and the mustard in the same row. And so those are a couple of combinations that we have just been recently looking at in our research, and particularly interested in how those combinations are affecting soil fertility, nutrient availability. And that intercropping system with pulse crops has potential benefits, in particular because as a legume, they will fix their own nitrogen as they grow together with the non-legume, the oilseed, and can also supply nitrogen to the partner as they grow. And I guess the other factor comes in is that legumes are great scavengers of phosphorus and other nutrients. They have that strong AM fungal relationship, symbiosis that develops there with those AM fungi in the rooting system. Now, a concept that's often used when talking about intercrops is something called the land equivalent ratio, or LER. And what the LER is it's the yield or the nutrient uptake of the intercrop partner one, divided by the yield of that crop when it's growing alone as a monocrop. Added on to that, the yield of the second crop in the intercrop system when it's growing as an intercrop, divided by the yield of that crop when it's growing alone as a monocrop. And when you add them up, if the number comes out greater than 1, that indicates a benefit from the intercropping, a more efficient use of that land resource. And so an LER value of greater than 1 indicates greater efficiency. It indicates synergy. So first thing we think about then would be yield in this system, and we do see greater efficiency in producing yield in these pulse seed oilseed intercrops as indicated by LER values greater than 1. For example, in our pea mustard intercrop system, our grain yield LERs for both the alternate and the mixed rows over a couple years of investigation that we had, and this was done by another MSc student, Melanie Reed, and working in my program, LER values that were greater than 1 for both mustard pea intercrop systems and also chickpea flax intercrop systems. So you say, well, where does that benefit in the intercrop system come from? Well, part of that can be related back to things like reduced disease pressure, which we saw in our intercrop system, less problems with leaf diseases when growing as an intercrop, also less disease infestation, but also in these systems that we've been working with, intercrop, pea mustard, chickpea flax. These intercrops that we've been working with are not fertilized. There's no nitrogen fertilizer. There's no phosphorus fertilizer added. And when we look at the LER values for nutrient uptake, and these are the sites we had a site actually on my farm at Central Butte and another one in the black soil zone in southeastern Saskatchewan, we can see their LER values for the chickpea flax intercrop system, whether growing in alternate row or mixed row systems, LER values that were typically greater than 1, indicating a synergy there in both nitrogen availability and phosphorus availability when those two crops were growing together as an intercrop compared to when they were growing alone as a monocrop. So we're seeing some enhancement there. And one of the sources of that enhancement that we were able to document using N15 as a tracer is nitrogen transfer from the legume to the non-legume crop during the growth period. And in terms here showing here how much nitrogen was transferred from the chickpea to the flax over the growing season at the different sites in the different years. It ranged anywhere from a nitrogen transfer of about 8% of the fixed nitrogen in that chickpea to upwards of 30%. So that atmospheric nitrogen that's fixed by the pulse and transferred to that non-legume partner certainly can explain at least part of the synergy that we see there in terms of increased yield, increased nitrogen uptake in these unfertilized pulse oilseed intercrop systems. The other factor is the AMF colonization, showing some data here. The chickpeas pretty well colonized, as you can see there to begin with, almost 100% colonization. But looking at the flax in those combinations, growing as a monocrop compared to an intercrop, particularly in that mixed row intercrop, we do see a significant increase in the percentage of colonization of the roots of the flax by the avuscular mycorrhizal fungi. So maybe that explains part of that enhancement of phosphorus uptake in that intercrop combination. So I think the biological nitrogen fixation contribution, the scavenging ability of the pulse crops, these beneficial microbial associations in the rhizosphere certainly are very important in contributing to soil fertility and health in organic crop rotations that we've seen here on the northern plains, and I anticipate would see the same in the type of thing in the southeastern United States as well. I think it's also important that we think about fertility in both short-term and longer term. The pulses are good scavengers of phosphorus, potassium, sulfur, but they will deplete soil reserves over time, especially if you're getting high grain yields and high removal then of the nutrient from the field in the crop harvest. So we also need to pay attention to long-term fertility management. If we're going to keep these systems going, we want to replenish the phosphorus, the potassium, the sulfur that we're removing with manure, compost, rock phosphorus, gypsum sources that are allowable sources of nutrient to replenish that nutrient removed in crop removal over the years in our organic cropping systems. So with that, I think I've used up my time, Dill, and thank you very much, everyone, for listening, the opportunity to participate, and always have to thank our funders for this work, no matter where we are. So thank you very much, and I'm happy to entertain questions and thoughts from the audience.
Thank you so much, Dr. Schoenau. This is really informative and lots of questions, and people actually, one of the questions, first one is, you were invited already for another talk. So people, all the participants, I think they were enjoying it, and especially your wealth and depth of knowledge of soil science and sharing it. I want to start the discussion with the global impact, because you have been doing so much of work on Saskatchewan and North America, but I don't know whether you heard about some of the countries in Asia, because I get this question all the time, but I wanted to pass that question to you. Start with discussion, because some of the countries with their economic recessions and their economic disparities in their countries, they wanted to move from inorganic system to the organic system. But the problem is they do not have fertilizer access. So if you are starting, if you're working in India or Sri Lanka or like that kind of third world country, how would they actually approach without having this fertilizer, kind of bring the organic cropping system? Like how do well?
So, I mean, I guess, what you're allowed to use in an organic system. I mean, if you're growing organic materials, organic crops, and you're selling those and you want to sell them as organic, you got to make sure that, of course, that what you're going to use as a nutrient source is actually allowable as a nutrient source. And that varies depending on the accreditation agencies. But, I think one of the best sources of nutrient and I've done a fair bit of work with over the years, is manure and compost. And if they are allowable, they are absolutely an excellent source of phosphorus, of potassium, even of micronutrients like zinc. And you mentioned about areas like India and Bangladesh, and crops that are growing there sometimes have some issues, and the population in general with zinc deficiency in their diet. And so through breeding efforts like you've been involved with, trying to improve the content of bioavailable zinc in a pulse crop is really important. And I think an organic grower there, as well as in other regions, may be paying attention to making sure there's adequate zinc in the soil because that will help increase the concentration of bioavailable zinc in the grain. So yeah, I really like manures and compost. They contain nutrients, nearly pretty well all of the 17 essential elements, not always in the correct proportion. And often we find our manures that we have here in Western Canada that come from cattle, high in available P, but quite low in available nitrogen. So that's where your pulse crop really comes in. It needs the phosphorus. It needs to get that from the soil or the compost amendment. Needs to get the potassium. Needs to get the sulfur. There's not much nitrogen in that manure, but luckily that pulse crop can fix it. And that's why we found it does very well in rotations with manure as the sole nutrient source. So that's the big one. Rock phosphate is another source. And I guess when it comes to rock phosphate, it's pretty insoluble, especially in our high pH soils. It's going to do better, especially if it's finely ground in soils like you'd find in North Carolina. But those pulse crops also, I think, can even do a better job than other crops of accessing that relatively insoluble phosphorus in that rock phosphate, because of how they can modify the rhizosphere and how they can develop these close associations with AM fungi that really impart a good scavenging ability. So I just think you got to put back in what you take out, Dill. Yeah.
That is a wonderful answer, because yeah, they are thinking about it because they don't have access to the urea or ammonium fertilizer. Only way. So, Dr. Schoenau, do they have to treat the animal manure when they add to the soil, or just they can directly apply this?
Well, I think always when you compost fresh manure source, it makes it easier to handle. It gets rid of the pathogens, gets rid of any weed disease issues. Good compost is great, but we've done a lot of work with fresh manure or just passively short-term compost, and it also works well as a nutrient source. It just isn't as nice to work with and handle.
Wonderful. This is really nice that they were wondering. And then there is the invitation for you from the Timeless Seeds to talk on February 10th, 2023. I'll copy this information and I'll send it to you.
So I see that's about weeds, Dill. So I'm not a weeds expert. So they probably need to find somebody else to talk about weeds. Yeah.
Maybe Dr. Shirtlift.
Yeah, that's right. Actually, that's right. And he's on sabbatical, so he may indeed have time. He would be a very good contact for that. Yeah, Dr. Shirtliffer, Dr. Willenberg here does a lot of work with seed destruction as well. Seed destruction practices that are non-herbicide oriented in organic systems. So yeah, they would be your better bet.
Yeah, and there's another question. Including pulses in an agriculture production system not only provide nitrogen benefits, but also provide the yield benefits.
Yeah.
And then the question, from your experience, what are the barriers in incorporation more pulses in cropping system? What are the barriers?
That's a great question, and I can answer that from my own personal experience, both as a researcher and as a farmer. And that what we've really come up as a little bit of a blockade, or not a blockade, but a bit of a barrier to continuing to increase the frequency of pulses in rotation, is root diseases. Root diseases, particularly Fusarium and Aphanomyces, and Fusarium, Aphanomyces root disease complexes, the Fusarium ones that affect both cereal and the pulse crops. We're seeing circumstances where we have some disease buildup in pulse diseases in some fields that are making it really challenging to continue to grow those crops without some type of chemical weed control. And even then, for some of the diseases that we have, like Aphanomyces root rot of peas and lentils dill, there really isn't even any chemical control. So we have to allow that. The only real control then is just to expand the rotation, the time period between when you grow a crop like peas from every third year or second year to every 6 or 7 years.
Right.
So that's been a challenge, is the appearance of some of these pulse crop diseases, particularly the root diseases.
So, Dr. Schoenau, can you a little bit explain to our growers about how they can have a nice crop rotation? Because in Canada, how we learned was every 3 years we grow pulses.
Yep.
But here, if you know, in South Carolina, we have several seasons because we can use the winter as a season, and spring, and then summer, and then the fall. Like, what is your recommendation for our growers to have a crop rotation in this kind of system, wet and humid?
Well, and I understand that you would be growing your pulses during the winter season, and
Yes.
that's a very good time, and as a cover crop, and as I think I would be looking there for pulse crops that have a high water requirement, that are going to keep that soil moisture down. Because my understanding is you folks, and you talked about a little bit earlier, to suffer from some oftentimes conditions of excess moisture at this time of the year. And that, from an environmental standpoint, it's that excess moisture that results in leaching and nitrate, that results in movement of phosphorus in water moving across the fields in the wet times. So that if you've got a pulse crop, is particularly one that uses a lot of water that we find, like faba bean, that can fix a lot of nitrogen and retain it, and maybe take up that phosphorus and cycle it through the residue. I think you're going to have a good set of conditions set up the following spring, that you won't have a lot of nitrate hanging around. You're going to have nitrogen, though, in that pulse residue, particularly in the roots, that's going to be released slowly through mineralization. And if that crop that was growing there in the winter took up some phosphorus, that's a good thing, because it's going to stop it from being washed off the field in the overland flow. And that phosphorus will cycle back out of the residue again. So, yeah, I think that's where I would see a particular fit of those pulse crops for your growers, is in that winter period, particularly thinking of it as a kind of cover crop that's going to add some nitrogen into the system. It's going to retain it and make it be used more efficiently. And not just nitrogen, but phosphorus as well.
Okay. And Dr. Schoenau, we are not doing much of the direct seeding, so we have to do seed bed preparation for the organic system to
Yeah.
control the weeds. So
Yeah.
what is your advice to our growers for the fall crop? Because we go as pulses in January. So what is your advice to make their bed preparation for the fall crop? What kind of cover crop you advise?
Yeah, and I guess weed control is always a challenge in an organic system. So you
Yeah.
try to keep that field clean. And there's maybe some strategies there, like some of the seed destructors that are used behind the harvesters that will macerate, or chaff lining, where you have the chaff of the crop concentrated in rows, and that helps encourage destruction or decomposition of the weed seeds. I'm also thinking about this intercrop system again, where you grow two different crops together, the pulse crop, and then maybe a cereal like rye in your case. And when you have those two crops together, especially if you got a competitive crop like a rye, that can help reduce the incidence of weeds in general in that cover crop. So maybe intercropping may be explored, particularly in your area, as a way of keeping down the weeds in that winter period when you're growing your pulse crops.
Thank you, because our growers are very much interested on what you're talking about, and because the intercropping is a new thing for us. I have a question regarding the intercropping. How do you harvest them when you have two different crops.
So that's the big thing. Of course, there's an extra step in the harvest in that after you've harvested your chickpeas and your flax, then you need to separate them out. So what you've got in chickpeas, you got a really big seed, and flax, you got a small seed. So actually, most of the organic growers that are successfully doing this, and there are quite a few here in Western Canada that are intercropping successfully, they have a seed separator that they use to separate out after harvest. It's an additional step, but also in this whole process, they also get rid of the weed seeds at the same time so that they produce a crop there that's a little bit easier to store and sell because they've separated it out. They separated the two crops. They've also got the weed seeds out of it as well. But that's the big challenge. Of course, in an organic system, you're not really worried about herbicides, but in a conventional system, there are some challenges in intercropping in terms of what's the likelihood of both of those crops being tolerant to the same herbicide. Although there's some pretty good products out there, because we've worked with that as well, that will control weeds, for example, both in chickpea and in flax. Wow. But I think in terms of an organic system, you're relying on the cultural aspect, the crop canopy, the competitiveness of the different partners in there, their ability to collectively use the resources more effectively, and thereby stop that weed or help prevent it from really getting a catch in the system.
Our growers love to hear this. Thank you so much. Now we want to move to phosphorus and other nutrients. This is a question from Dr. Emerson Shipe. He's a soybean breeder at the Clemson University, and he's now retired and working with me on pulses. So his question is, do you have any thoughts or suggestions on maintaining necessary phosphorus and potassium soil levels for growing organic pulses in southern U.S.? Because this is one of the big issues for us.
Yeah. And I think if you can get an organically acceptable compost or manure, they are excellent sources of phosphorus and potassium to replace that which is removed in crop harvest. Rock phosphate. The pulse crops are pretty darn good scavengers, and I've been amazed on my own farm in our trials how we can continue to grow pulse crops on very low phosphorus fertility soils. Potassium deficiencies, not a big issue in Western Canada. Our soils are quite rich in available potassium, but there for potassium, I really think your manures and composts are also really good sources. Again, pulses are good scavengers of soil potassium, and I know in trials that we've done, relatively little response of pulse crops often to fertilizer K because they do such a good job of getting it out of the soil. But over time, you are going to deplete those reserves, and so you do have to think about how you're going to put it back in. And again, I come back to manures and composts. I think an excellent way.
Yeah, I agree with you. So one of the projects that done by one of our PhD students, so we found one of the phosphorus is the most limiting factor in the organic system.
Yeah.
So, and even though we can add the starter phosphorus or starter nitrogen with our seed, it's difficult because we have sandy soil and it leached out and it is not going to be available. So one of the things that's why in our program we thought of breeding for low phosphorus cultivars in the organic system, reduce adding fertilizer. Because most of these growers, Dr. Schoenau, they want to add less input. Yeah. So that's why they wanted to have pulses. But we are running into problems with inoculum. But you said that we have to find the exactly proper inoculum which is suitable for each crop.
Yes. Yep.
Do you have any recommendation for growers because they have issues with finding because they are different inoculum, peat based or liquid based? What is your recommendation on those inoculum for farmers to buy?
What we found here is that generally the granular inoculants are at least as good as any other of the application strategies for inoculant, and sometimes they're better. And the reason why in our environment is that with the granular inoculant, you can put the inoculant down deeper into the soil, and that encourages the development of nodules on the lateral roots rather than just staying up close to the surface. And the trouble is, especially in our environment, which is a dry environment, if you get all your nodules formed on the roots that are close to the surface and it gets dry, that really negatively affects those nodules. The granular inoculant can put it down deeper in, closer to the deeper where the deeper roots are going to be. And that, especially under dry conditions, seems to give an advantage in nodule viability and functioning. Now, whether that would be the case in South Carolina would be interesting to do as research, a comparison of different inoculant formulations. We have issues here with some of the seed applied ones, in the sense that a seed applied inoculant, especially a liquid inoculant, if it's allowed to sit around for a long time, more than about 24 hours, the viability of that a seed applied inoculant goes down. And if it sits in the back of a truck for 2 or 3 weeks, we often recommend the grower go back out and inoculate again. But I think an interesting thing that we're also observing is that over years, as we've been growing these pulse crops, we are seeing less response to inoculation in general because we are building up a population of those soils, or a population of those rhizobia in our soils. But still, we recommend growers inoculate with a commercial inoculant every time they grow that pulse because of loss of genetic purity of that organism in the soil over the years if you just leave it there, and also that you've got that inoculant in the soil, that rhizobia is going to be close to where those roots are.
That is wonderful. Our last question, Dr. Schoenau, we can let you go, but we have one more question. What is your recommendation for our growers as a starter nitrogen? Because, our soils are really sandy. Most of in Pelion, South Carolina. When you visit here, I'll take you around. So what is your recommendation as a starter nitrogen for the legumes?
Yeah, so I just completed some research work on starter, and this is fertilizer nitrogen for the pulse crops. And I will say that in general, we did not see much response to starter fertilizer nitrogen in crops like peas, lentils, chickpeas, faba beans. We did see some response to starter nitrogen in the soybeans because it takes longer for that nodulation process to get going in soybeans compared to other grain legumes. But honestly, I think our conclusions of that work were that really, there was no benefit to going above about 10 pounds of available nitrogen per acre as a fertilizer added as a starter. So just only very small amounts of starter N, if any, seem to really be needed in our environment.
Okay. So because of the chickpeas, they could not harvest it. Plants were really small, and because they did not fix enough nitrogen. So that's why the growers were thinking like, okay, let's add a little bit of nitrogen. That's what I recommend.
Okay. And I think on those chickpeas, I would doubt that there is an indigenous population of Rhizobium cicer. And so I think, it's just like when we started to grow soybeans here in Western Canada, never ever growing them before, started about 10 years ago, you had to inoculate those soybeans with a Rhizobium inoculant, or you got very, very poor yields because there was no other way for the plant to get nitrogen. And there was no indigenous population of the Rhizobium specific to the soybean.
Okay. One last question. I'm not going to ask any more questions. Have you had any experience on applying AMF inoculant on lentils or peas? And have you seen any benefits on the pulse crops?
I have not done that specifically as a principal investigator in my own research work, but I have been involved in some other studies, some other graduate student projects where they have looked at applying AMF inoculants on these pulse crops. And I would say that the results are mixed. I don't think any of the studies really showed any hugely consistent positive benefit from the inoculants that they evaluated. And I'm thinking of one PhD student in particular really didn't see a big benefit from the inoculants in our soils. But that could also be because, in general, as I showed in my slide, we get a lot of AMF colonization naturally from the population in the soil to begin with. So maybe in different environment, different soils where you don't have that high indigenous population, that high capability, there you may see a response. But we really didn't here see much of a response still.
Dr. Schoenau, thank you so much for sharing your wealth and depth of information with us, and our growers, scientists, students. They'll be happy, and that's why you got a lot of questions, because they were
Well,
so
great, great.
happy
No,
to have your knowledge. So I'm hoping to invite you to South Carolina, go around with our farmers and actually tell them more about what you're doing. And
yeah, and
I'm
I'd like to see. It's nice to see firsthand exactly what's going on, and certainly comparing and contrasting to my own operations and farms that we have here in the northern Great Plains. So thank you very much for having me,
we
and
will
that's great.
we will be in touch, and thank you so much. And thank you all for joining, and we will see you again next month. And our speaker, actually another wonderful Canadian researcher, Dr. Susan Whiting, and we're going to talk about benefits of eating pulses. So we manage the soil, we grow the crop, and then next webinar, we're going to talk about how it's going to be nutritionally benefit. Thank you all again. We will hope to see you February 22nd, 2023. Thank you. Goodbye.